could you?
All right. It's my pleasure to introduce our next speaker, Julian Berro at Yale. Julian is a rehabilitated mathematician. As someone that tried to do experiments just once in my life, and I broke every machine and contaminated samples and was chased out, I'm amazed by people that are able to span both, and I think you'll be impressed today too. Hey, Julian.
All right. Thanks a lot for the introduction. That's exactly the perfect definition of me. Rehabilitated mathematician. What saved me in the experimental world is I work with yeast. I've worked with yeast almost exclusively up to this point, working on endocytosis in yeast. And I managed to get funding up to this day and get tenured even to this day. So there's hope for everyone who wants to start experiments. And yeast are nice because they're very forgiving. And it's a very clean system and very simple system. So it was simple enough for a mathematician like me, but it's still quite complicated. Okay, so today I'm going to talk about the fantastic forces and where to find them. So where to find them, the answer is everywhere. So at the end of the talk. So we've seen a lot of talks that talk about forces. I don't have to introduce the things. This is not from my lab. That's why it's so beautiful movie. And so cell motility, adhesion, cell division, those are the macroscopic effects. There's a lot of forces in membrane trafficking, in intracellular processes, and so on. Also important in development, right? So just to, I don't really have to set the stage here. I think everybody's aware of that and agrees with me on the forces are everywhere. Now the question is how to find them and how can we measure them? So how can we measure the forces? And it's at the molecular scale. That's what's missing. You're gonna have to guess. It can be fun. So one way is to compare to pre-calibrated forces, right? So I like to make this analogy, measuring weights. You have different ways to do it. One way is if you have a preset, if you have a pan balance and a set of pre-calibrated weights, then you can compare the object you're trying to, for which you're trying to measure the weight, like the apple, you put it on one, you pick a weight, a reference weight, you put your apple on one of the pans, your reference weight on the other one, and you can tell if the apple is lighter or heavier than the reference, right? You keep doing that for different weights and so on and so forth, and you can basically narrow down the weight by dichotomy process, right? That's a word I realize is I got from my math training. Dichotomy. Biologists don't understand what dichotomy is. But anyway, so I call it bracketing now. Anyway, so that's how you get there, right? So how can you do that in at the molecular scale? So it happens that cold coils, so two alpha helices that are wrapped around each other, are very great tools for that. For one, is they unfold under a specific force threshold. They can be tuned thanks to a lot of research from 40, 50 years ago. It's pretty understood, well understood how they're stable or not. The nice thing is the end-to-end distance can be very small. So 1.5 nanometer, it's about a few amino acids if they are in a flexible chain for the end-to-end distance. And the other nice thing is you can play with what's in the linker here, and doesn't change the stability of the full coil. So I brought a prop, which is basically a Velcro. That's from my, I stole it from my daughter with a taekwondo, because that's our taekwondo belt. So there's a Velcro here. And so the idea is if you pull not too much on it, nothing happens. And if you pass a certain threshold, it opens completely, right? And that's it for now. So The nice thing is we can actually do the same thing with the col-col, but with optical tweezers. So you attach the col-col between two beads, and then you do cycles of pulling and releasing, and you get this kind of curve. So it's a force extension curve. So if you follow the black curve first, so you have the col-col that's that's folded at low force, and then you keep pulling, and then you go up that curve. And at some point in this region, there's a little bit of flickering, and it's basically because the col-col opens and closes. And then you keep pulling, and then you have an open col-col, and then you stretch it. Okay, and then you release, and you follow the red curve, you go back, and then it refolds all of a sudden to the end, right? And so those are the tracks from the same col-col that's over several cycles of open and pull. I splayed all the tracks here, so the absolute extension doesn't really mean anything. It's just so they don't all overlap on the same, but they're all very reproducible, right? And so before I forget to acknowledge the people who did the work, Yuan Ren was a postdoc in my lab, who's done really most of the work on this project, and has got a lot of help from Jie Yang, who was a postdoc in Yongli Zhang's lab, with a collaborator at Yale, who's a specialist in optical tweezers. Okay, so from there, we basically developed a series of col-cols that are references from 3 piconewton to 20 piconewton, which covers a pretty decent range of what the forces at molecular scale you would expect to see in vivo. So now, how do you use them in vivo? Because you cannot detect in vivo such small openings. It's going to be just a nanometer scale. So what you do is if you have your favorite protein of interest, say it has 2 domains, D1, D2, you basically graft your col-col. Basically, you genetically encode the sequence for the col-col between the 2 domains. And what happens is there is not much force. It stays like that. The protein is usually fully functional. When there's enough force, it opens. And you remember I was talking about this linker that we can play with what we put there. And it happens that we put there residues that bind to something else. Right? And so basically, we can recruit something only when the col-col is open. Right? And so one of our favorite examples is the split GFP. And so it's basically split GFP. If you're familiar, the GFP is a beta barrel with 11 beta strands all around. People have engineered in the past split GFP, so it only has a first 10 beta strand. And the 11th beta strand is basically not folded on its own. And that's what we use as a linker here. And only when the col-col opens, then it can recombine and bind to the GFP. So it's really this idea that when the col-col is closed, the reactive residues are kinked here, and so you cannot really bind to the GFP. And so only when it opens, now you get accessibility for this split GFP to come and bind, right, and become fluorescent. Okay, nobody laughed at that job. Anyway, so
simple and clear, so nothing to laugh at.
Tough crowd. But the nice thing is basically you get no signal when it's when the col-col is closed, and signal that basically localize where your protein of interest localizes. So here, our protein in this example, they're little puncta, which are basically endocytic sites. Right, so that's basically the somewhat serious animation of what I was showing. And we got, so the nice thing with split GFP has very good on to off ratio, The downside of it is it's a little slow to mature, and it's also somewhat irreversible. It's not completely irreversible. When we say irreversible here, it's the off rate constant is in the hour on the hour time scale. So for the processes we usually care about, they're irreversible. So that means that we can use them as force recorders in the sense that we can tell that the force has happened at some point, but we cannot tell when it happened and where it happened. For that, we developed other readouts, and it's all by exchanging these linkers. And we have different systems where we have basically a GFP that has a little tag on it, and I can bind to a complementary tag on the linker. And so what you do here is you basically relocalize the fluorescence to your site of interest, right? So the downside is you have relatively high background, but you get better temporal resolution, right? And with the system we've played with, we have some systems that are in the second time scale for exchange, right? So that's pretty good. Okay, we developed, so that's what we call the force live reporters. And we developed different readouts for different applications, including split luciferase assay, TV cleavage, and protein condensation that is sometimes convenient. All right. So that's our nomenclature now. We use the Chinese symbol for force, Li, and the underscript is the force threshold, and the upperscript is the readout, right? So you're going to see that nomenclature in the rest of the talk. Okay, so we've been applying the true reason we developed these tools, and also that's another motivation why I started doing experiments, is because when you collaborate with experimentalists and you do modeling, it's sometimes hard to get the experimentalists to measure what you want them to measure. It sounds like familiar, right? And sometimes you want to measure things that are not measurable. And so the biologists don't necessarily want to spend their time developing new tools to measure things that they don't care about so much. So basically, you do it yourself. So that's how I ended up developing these tools that were all about, because I was working and I'm still working on endocytosis, and the whole point was about how forces are produced and sensed during endocytosis, but nobody had a sense of how much force was there. So endocytosis, for the ones who don't know, it's basically the process of going from a flat membrane to a vesicle, and you just need to deform the membrane. What happens here is you actually have to work against a few things: membrane tension and turgor pressure. And it happens that in yeast there is a very high turgor pressure, about 10 atmospheres, which is really the driving force you have to go against when you do endocytosis. And so nature evolved a nice process that's all based on actin that's conserved across eukaryotes to help produce the forces. I don't want you to think too much about all the details, but you have all the usual suspects in actin systems. So it's all driven by apto-free complex branched actin network. And basically, the whole point is that the actin polymerizes, is nucleated at the base, polymerizes go back through the tip, and eventually it's pinched off, right? What I want to stress on here are the time scales and length scales. And that's what makes it probably the most different from pretty much all the talks we've seen the last couple of days. So it's all very fast, 8 seconds, and very small. So it's under the diffraction limit. So you cannot just look at things to know where they are. You have to be a little creative. So it makes it a very fast, transient, diffraction limited out of equilibrium system that makes it fun for someone with a quantitative background and and where mathematical modeling and quantitative analysis could be very useful. So that's the best you can see when you tag a protein with a fluorescent tag. That's what it looks like in yeast. And I forgot to mention, I work with the best yeast on Earth, which is the Pistosaccharomyces formidae. And so it looks like that. So every dot you see is basically an endocytosis event going on. I'm going to play the movie. You can pick any spot. Try to pick a spot that's on the side, not at the tips. For some reasons, I'm not going to talk about endocytosis is usually polarized where the cell grows. But look, so it's pretty busy at the tips, but look on the sides. And any spot you look at, you're going to see something going on. And so what you see usually is a spot that appears, the intensity goes up. And then basically when the intensity is maximum here, it starts moving around, right? And basically it's this process of assembly of actin, and then it's pinched off, right? And here it's an actin ferment cross-linker marker. And it's inverted contrast, so the more black. So, oh yeah, I wanted to do that here because it's going to be the only thing that I found is relevant to machine learning in my talk, and I apologize for that. But we spent a lot of time when I was a postdoc early when I started my lab, tracking those patches, extracting a number of molecules from them by calibrating the microscope properly, and then making models out of it. We're still interested in doing that, but that's, funding is tight. So that's not the lowest hanging fruit we can go after these days. So it's a little bit on pause. But basically, the machine learning aspect here in tracking, we spend a lot of time manually tracking those things because there's a lot of overlap, a lot of problems, even with the nice tools that Guidance and Jacquemin have developed in the past are great for that, and that's what we've been using. But still, after that, we still need to do a lot of post-processing, and that's something I always thought machine learning would be very good at for us. There's also a problem of realignment of time because there's no absolute time here, and so if you get different tracks, you don't necessarily know how to realign. And the other problem is it's relatively low temporal resolution. But in principle, you can get a higher temporal resolution by using the right algorithms of machine learning. And it's very, very similar. Started implementing that. It's very similar to the algorithms that I used in single particle cryo-EM reconstruction. It's the same concept of very noisy, low resolution data that then you can realign and get back to a higher resolution than what you get at the beginning. And then there's also a lot of opportunities for machine learning tools for model selection and parameter estimation, as we've seen in the last couple of days. Okay, so how the forces are produced, I touched on that a tiny bit. So that's what we call a push-pull model in the sense that actin is nucleated around the pit, and basically it's going to polymerize, but instead of, so it's really exactly the same concept as for cell motility and the lamellipodia, except that in the lamellipodia, as you saw yesterday, there is a bit of retrograde flow and some movement forward. Here it's only movement as a retrograde flow. And so it goes backwards. And it happens that it's not polymerizing here. And you have these little red sticks here that are basically adapter proteins that basically link the tip of the membrane to the actin cytoskeleton. So it's pushing on the side. Because of retrograde flow, it's pulling in the middle, right? So the field calls it push-pull model. Anders Carlson has an even better way to call it. He calls it weed pulling model because you have a weed here. And you grab the weeds and then you push with your legs, and that's how you pull the weed out of the ground. And I really like that analogy. So your legs are the actin and your hands are the adapter proteins.
Sorry, what I didn't get it. What does the pooling?
So the pooling is really actin polymerization from the base, and sorry, the pushing and the pooling are these membrane membrane binding proteins that connect the tip of the membrane to the actin.
So you basically grab the weed and push with your legs, right?
Yes.
You're not pushing with your hands.
Yes, exactly. Yeah. You're pulling with your hands because your legs are pushing, right? But not because you put any force on the hands. Okay, so the question was how much force is needed to do that. So there was no tools to really measure it. So brilliant theorist in my lab, Rima, with now at Xiamen University, has his own group, did some theory with using Helfrich theory of membranes, used some published data, EM data from Marco Kaxonen's lab. And basically, we were trying to play around with the different assumptions, hypotheses, conditions to reduce as much as possible the force. And we managed to get a force height relationship like that. Usually in pure lipid systems, you have a big peak here at the beginning, which means that it's very difficult to do the initial step of deforming the membrane. And once you have something that looks less like a tube, you can elongate at a constant force, right? And so with some work that we did, we managed to reduce that threshold. I'm going to pass on the details, but the bottom line here is you need about 2,500 piconewton of force. That's really the best we could do using an idealized theory for the push-pull model, right? And so basically it's the best case scenario, right? And so the question was, can we produce, are 2,500 piconewton produced for real, and can we produce food? So are they produced? So basically now we have the tools, the force sensors I was talking about. So one of the two adapter proteins, the red sticks I was talking about, is called ENT1. So it has a membrane PRP2 binding domain, actin binding domain, clathrin binding domain. So we put the sensor between actin and lipid binding domains. So where they are here. And so that's the control where we should expect if all the sensors are open. This is what you expect when you put the 3 piconewton sensor. So now you can tell that the force is larger than 3 piconewton. And that's what you see when you have the 5 piconewton and what you have with the 10, right? So it's mostly off. You can see a little bit of signal, which basically tells us that probably the force sometimes goes a little above 5, but it's very rare. But definitely you get no signal with 10, right? Which means, so the nice thing with these sensors is they're very self-controls for each other, right? So if you get the 5 has no signal, the ones that are higher threshold should have no signal or less signal. Gotcha.
Measurement of the force on one adapter, or like the total force on the whole kind of bud.
So it tells you the force on one molecule.
How much?
So, and then we're going to get back to that after, because it doesn't tell you the force on you don't know how many molecules are under force with just that measurement. So the conclusion from here is that the force on ENT1 is about 4 piconewtons during endocytosis. And then we moved on to the other adapter protein called ENT4. And so it's conceptually the same, membrane binding domain and actin binding domain, but it's a little more complicated than what's going on in the middle. And we did the first measurement that's really high, 19 piconewton, close to actin. And when you think about the range of forces, for example, actin polymerization alone can produce, or molecular motors. It's in the low piconewton range, right? So 19 means that there's a lot going on from the whole actin network that basically integrates force in some ways to transmit 19 to this molecule. So we are pretty happy, and then we measured. Originally, we measured that as controls. We moved the sensor at different locations. We were really not happy because the force is 19 here. It's 9 close to the membrane. And we're like, what is going on? Because we would expect, if it's like a rope, that the force at one end is the same as the other end, right? We did absolutely all the controls we could imagine. And we still got that. The thing that reassured us to some extent and finished to convince us is it's kind of decreasing. Wow, we followed up on that. And we're like, maybe there's something else going on, and we're going to discover new biology from there, from these force measurements. And so the first follow-up discovery was to basically rethink a little bit our naive interpretation of how the adaptive proteins work with each other. And we basically considered them independent. It's like this protein binds to actin, this one binds to actin, and it's a rope between the two. But actually, there are other domains of these proteins. They can interact with each other and bind to each other. And so there was this idea of a force redistribution system, right? And so how can you test that? So it's pretty simple. You basically mutate the actin binding domain from one and see if you basically see more force on the other one protein. So that's what we did. And so here you're mutating the actin binding domain of N4, and the 19 piconewton disappears. So basically, we're not binding actin anymore. All the force disappear up to very close to the membrane, just before the protein-rich domain here. So basically, what it tells us is there's something around this region that is attached to something else that transmits the force, right? And you can do this. You can look at the force now on N1 in this mutant that doesn't have actin. And basically, the force on N1, remember when I was showing it's 4. Now it goes up to 10. Right? So basically, what it tells you is if you don't transmit the force from here, you get more force from the other proteins. Right? That's really the idea of redistribution. You can do the other way around. So if you mutate the actin binding domain of N1, then you get larger forces on N4. So I call it the cathedral model. It's really the same idea of redistributing the forces from the roof to many different pillars. Right? Now with these numbers, is it enough to deform the membrane? Remember the theory was saying you need about 2,500 piconewton. So you get these numbers that are closest we could think of. And now if you estimate, again, best case scenario, from previous work, we counted the maximum number of N4. We guesstimate the number of N1. We didn't do the exact measurement, but it cannot be much larger than that. And you do the math, and at the end, you get about 2,000 piconewton. And that's still less than your best case. So this is the best case scenario, and it's less than the worst case scenario. I don't know how to, the best case scenario. So basically, you're still, even if you put yourself in the best conditions, you're still less than what you would expect to be reproduced. So basically, it tells us that this push-pull model is incomplete or just plain wrong, right? And that's something we're working on. Okay. So another fun discovery, and depends how much time I have.
Enough.
Yeah.
5 more minutes.
Okay, very good. Another fun discovery was this thing, because here, as much as I could understand that here it goes from 16 to 11. There's a lot going on here. Calls, potentially binding to other things. Here I understand the drop. Here I didn't understand because it was admitted to be nothing, right? Just disordered. And that was just before AlphaFold came out, but we had a little bit of suspicion, so we purified that region. And it ends up to be a domain that's a 5-bundle helix. So we purified that domain, and AlphaFold predicted it as a 5-bundle helix. So 5 helices, and we do the same thing. So here it's you're going to have to follow the red first. So again, force extension curve. So you attach your domain between 2 beads, you pull. So this is basically the stretching of the linkers that attach them to bead. And then here there's a clean, clear jump. There's not even flickering. And that's very big. And then you keep extending. And that basically tells you that this 5-helix bundle unfolds in one step altogether. It's not like one helix at a time, right? The nice thing with this is, the interesting thing with this is the force drops a little bit, but the extension becomes very big too. It's more than 20, 30, 40 nanometers, which is basically the order of magnitude of what your endocytic site is, right? If you remember, 15 nanometer is the size of the vesicle, right? And it's going to be important in a second, right? So that's the first result. The other thing that's different from our sensors here is, you remember the sensors, they would go back, refold exactly at the same force as they unfolded. But here you have to really go all the way down to almost undetectable force for the whole bundle to refold, right? And then you can redo a new cycle, and it works beautifully again, right? And so it's a really cool system here because you have an hysteresis system. So basically, you can bear force up to about 15 piconewton. At 15, you basically break, and to go back down to 0 piconewton to refold and go back into the cycle. And it happens that it ends up being a little mechanical circuit, and something I don't talk much about here, but we and others previously showed that this actin binding domain is also a catch bond, right? So it grabs stronger on actin filaments when there's more force on it, and so you get this this little 2-domain system where basically you can exert force up to 15 piconewton. At 15, this domain unfolds. And then because the end-to-end distance becomes very long, what we think is going on is basically the filaments can't even pull the whole thing. So the consequence is really that the force here becomes very low. And because it becomes very low and it's not catch bond anymore, it's a catch bond system, it falls off, right? And so basically you have a way to regulate the force there. And so you can do a little bit of modeling and test the different hypotheses. So if you have or not this domain that unfolds, have or not hysteresis, if you have a catch bond or slip bond here, you get different results. And here you get this kind of input force, output transmitted force kind of graph. And basically what we're showing here is this green region is basically a force that's in the 5 to 9 piconewton. That's output force. For an input force from actin, that would be from 5 to 30 piconewton. So you're really smoothing the force transmission there. And the other thing I wanted to point out for here, some adhesion people in the room, the reason we call it R end is because it looks, 5-helix bundles are what you see also in thalin. And the touch is also the same kind of domain, it's homologous to the one you see in thalin. So it's basically a mini thalin, and for the mini thalin, and it really works the same way. So it has no specific homology to taline. It just works the same way. And R is because the rod domains from taline, N is for, we say it's for endocytosis, but I usually say it's for REN domain. All right. I'm going to skip that, just giving you the result, but 9 piconewton is super high for non-transmembrane proteins, so it should be stripped of the membrane. And it happens that almost 10 years ago now, we had the data that proved that, but we didn't really know about it. And it was basically single molecule data that shows that N4 actually stays, basically moves the same way as actin on these systems, right? And we're very confused about it, but it's probably because it ends up being stripped from the membrane. So that's our new model. Instead of push-pull, it's push, redistribute, pull, strip model. It's not a good name. Or the slippery, intricated wheat pulling, because you slippery or ponytail model. If you've made a ponytail in your life, you understand what I mean. So basically, you have actin polymerizing from the base. There's going to be a retrograde flow. It's going to be caught by these adapter proteins, produce up to 19 piconewton of force. Because of the buffering, there's only 9 piconewton that transmitted to the membrane. And then you elongate a little bit the membrane, come off, and all this process from our single molecule data, we know it's as fast as 1.5 seconds on average. And basically it repeats about 5 times to do the whole cycle to elongate the whole invagination and pinch it off into a vesicle. Right. The ponytail is because when you do a ponytail, you never do it in one shot, right? You have to do like that, right? So it's the same thing, like because you don't catch everything. So to do a ponytail in one shot, you need to be super precise, right? But you can be not too precise if you give yourself a few times, 5 times to kind of put all the hair in the back. All right. So that's pretty much it. So those are all the projects we are working on these days. So I showed you a bunch of things on endocytosis. We have collaborators for exocytosis. We're getting into the adhesion business. We have a preprint on formin CDC12, which is a formin for cytokinesis in yeast. There's some fun results there too. Collaborating, and actually all our funding these days is on CAR T cells therapies. Don't ask me how I got there. And some nuclear envelope and chromatin proteins. Just because there are some adhesion people in the room, I wanted to show the results on vinculin, and because it's kind of the gold standard in force measurement. So fret-based sensors, so forces are in the 3 piconewton range. With our sensor, so we image a vinculin. So that all the vinculin is in red, and the vinculin that's on force that's above 3 piconewton is in green. So it matches in terms of pattern, but the intensities don't necessarily match, right? So not all vinculins are under force. We can do 2 colors at the same time. So we can get the 3 piconewton and 5 piconewton in the same cell. It doesn't render so well on this projector, but basically you get more fluorescence for the 3 piconewton than for the 5. So basically it tells you that only a small proportion of, someone wants me to shut up. A small proportion of vinculin that's on high force threshold, and we could quantify that in some ways. And basically that's 3 piconewton, everything is renormalized to 3. So we have a way to get a sense of what's the proportion of vinculin under force to total vinculin, but we didn't do it because we didn't calibrate properly things yet. So we don't have that number of, that absolute number, but basically if you renormalize everything to 3, almost 90% of the vinculin that are under force at 5 piconewton, and then not too, like about 20% at 7, and only 10 or less at 10 piconewton. So some vinculins see force that are quite high. And with that, I want to thank again my lab, the fantastic people from the lab who work on the fantastic forces, especially Yuan Ren, who is now assistant professor at Beijing Normal University, and all the collaborators and funding while it lasts. Thank you.
Take one question before the coffee.
So the threshold 2500 piconewton, is that from pulling a planar membrane or a curved membrane?
Right, so it's planar membrane and it's a point force at the tip. It's kind of best case scenario.
Might be lower if it's from pulling the curved membrane.
We can talk about that. In our case, I don't think it would change, but this is a we can talk about it.
You want to take?
Yeah, yeah. I mean,
one more. Yeah. There's someone eager over there. Oh, yeah, or yeah, go in there.
Whoever wants to go first. Go ahead.
Oh, this is a naive question coming from an unrehabilitated mathematician, but I could imagine that production of new membrane or exocytosis would relieve some of that tension force. Is that
right? So
significant?
Yeah. No, it's very good, very good question. So in you're absolutely right. This is what happens even in neurons at synapses when there's fast release. There's also fast endocytosis to balance membrane tension. This is definitely a clear effect. In our case, where we don't care that much for yeast is because the main source of things you have to work against is turgor pressure. Membrane tension doesn't influence too much the results. But this is a very good point, yeah. One last one.
Really beautiful talk. I was just curious about the force balance, and I'm not sure if I'm getting it right, but would it be possible to have, so one thing that the process is so fast that you actually need that force imbalance for the process to go forward? And second question was, could there be some viscous dissipation as well? Here you're assuming that the forces are should be in balance all the time, but could there be some sort of viscous dissipation happening, which
I see what you mean.
which would lead to force imbalance, like different force being dissipated on the other side?
Right. So you would imagine that yes, that you're in a viscous system, and so you pull hard here.
Membrane is fluid enough to dissipate. We don't think it happens. One of the argument I would give is because of low Reynolds numbers. Like the speed, your first part of your question, I don't think think for that argument would probably, there's no inertia. So I would say we're safe on that end. For the thing is, that's true that there is, and it's kind of, let's put it this way. The way I would see the viscosity of everything, the way I interpret it is binding to other things. So there's a apparent viscosity, but at the molecular scale, it's more binding to other things. That's what I would guess. Now at equilibrium would just, if it was just pure viscosity of the media, would that dissipate? I don't know. I mean, Chris seems to come to my rescue. I don't know. I don't think so, but that could be wrong.